So, pathogens. For examples, the SARS virus, the plague, also known as the Black Death, smallpox, and the Spanish influenza, 1918 to 1920, killing between 50 million and 100 million. Yes, that's right. A huge killer. H1N1 virus. That's why when swine flu kicked off a year ago, there's a lot of concerns around it because its biology was very similar to that of swine flu. They can be bloodborne, waterborne, airborne, saccharophilic pathogens. What pathogens do is basically there are disease-producing agents that causes serious infections, like bacteriums, virus, microorganisms, and they infect the host, the vertebrates, etc. Us. So it breaks down your white blood cells, your immune system, and also it can destroy your platelets as well, which is what's in your blood to help sort of clot any open wounds that you may have. So basically it can cause serious hemorrhaging. I've been meaning to do this video for quite a long time on retroviruses and why they've come about and why they still exist today and throughout our whole genome. But some great analysis have been done recently from virologists. I've read plenty of journals on this. So how this happens is these viruses basically pass on their genetic material through replications and leave behind these mutations throughout the genome. These virologists have been analysing open reading frames whilst looking at the genome, uncovered that humans and the vertebrates genome also contained many ancient sequences, from Ebola and Bornaviruses, two deadly virus families. Because neither virus family insert their genetic material into the host genome during replication, like retroviruses, the discovery was almost unexpected. Specialists compared over 5,600 viral genes from all non-known retrovirus families, a single-stranded RNA genomes, to the genome of 48 vertebrates, including humans. They uncovered 80 separate viral sequences integrated into 19 different vertebrate species. Interestingly though, these viral sequences from ancient relatives were just two viral families, Ebola and the Bornaviruses that I mentioned earlier, both of which cause hemorrhagic fevers and neurological disease. In a nutshell, these viruses are RNA viruses. Basically they replicate the RNA, but there's no mechanism to produce DNA. Have they no mechanism to get their genetic material integrated into the sequence of the genome? Some of them don't even enter the nucleus when they replicate. During normalization of replication of genes is when a chromosome forms two chromatids prior cell division. Its constituent DNA also replicates. The component strands of each double helix unwinds, exposing the organic bases on the nucleoplasm. In the nucleoplasm are the corresponding bases attached to sugar and phosphate molecules. The combination of organic bases of sugars and phosphates are called nucleotides. The bases of the appropriate nucleotide become attached to expose bases of the separated DNA strand, and so it builds up on top of the molecules of DNA. Exactly the same sequences of bases and its original partner. In this way, the genetic code is inserted intact and passed on in the chromosomes to a new cell. These sequences, some which may have been integrated into our genome over 40 million years ago, as I've already mentioned, have been largely concerned over evolutionary time that suggests to scientists there's a host of selected advantages, perhaps protecting species, protect them from future infections by viruses from those families. I'll give you an example of a modern day scenario, sickle cell anemia. What has happened here, natural selection has driven Africans and certain parts of Asian people to protect them against or kind of vaccinate them against a parasite known as malaria and the most lethal Plasmodium falciparum. What this analysis shows is integration of this ancient viral sequence has been mediated by different events of lines of measurements which is abundant in the mammal genome. these virologists have clearly demonstrated conclusively that viral sequences of the same biological function that take a bit of understanding going forward. But when you're looking at some of the expression of these virals in the open medium frames, i.e. human tissue, does support the possibility that biological things are still active in ourselves. As you can see, how some virals and that only have basic early life forms only have RNA, and there's no genetic makeup to produce DNA. And with this news, I'm coming to you today. Only just recently, in the last few days, there's been more evidence or basic early life, bacteria life, possibly being found on Mars. So it is kind of watch this space, and I will bring this suggestion up to Professor Mason at our meeting in a week's time, and to see if he has any more on this. But it does look like there is bacterial life that's been found in some of the rock formations on Mars. It's got SETI, Search for Extraterrestrial Intelligence, quite excited, as well as NASA and so on. So top organisations are looking at biology, astrobiology. The question is, if we can get some samples of these bacteriums that are in these rocks, no doubts they would have followed the same evolutionary path as ourselves. Unfortunately, for one reason or another, life just didn't take hold like it did on Earth. Anyway, thanks for watching. Lots.